All right, good morning, good afternoon. Welcome to the environmental lecture presented by the Environment Group of American Chemical Society Pittsburgh local section. And this lecture is co-sponsored by Calgon Carbon Corporation, a global leader in active carbon products and solutions. And I'm your host, Ron Hong Lin, a member of ACS Pittsburgh local section executive board and chairman of the environmental group. The environmental group is a topic group within the local section, formed with the aim of educating and promoting awareness about our environment. We support various activities, including this environmental lecture series, and which we intend to continue monthly with speakers who are experts and leaders from a wide variety of chemistry and environmental science backgrounds to promote a deep understanding of science, technology, and our environment. Just to let you know that this lecture is being recorded and will be available online later. I'd like to remind the attendees and future viewers that the views and opinions expressed in this and any future lectures are those of the presenters and do not necessarily represent the views of the ACS Pittsburgh local section. Now it's my great pleasure to introduce today's speaker, Dr. R. Paul Philp. Dr. Philp is an emeritus professor of petroleum and environmental forensic geochemistry at the University of Oklahoma. He received his BS degree in 1968 from the University of Aberdeen, Scotland, and a PhD in 1972 and a DS in 1998, both from the University of Sydney, Australia. He was an associate research chemist at UC Berkeley, and later on principal research scientist at Commonwealth Scientific and Industrial Research Organization, or CSIRO, Sydney, Australia, before joining the faculty at the University of Oklahoma in 1984. Dr. Phillips' research interests include petroleum, environmental, and forensic geochemistry, with an emphasis on molecular and isotopic characterization of oils, gases, rock extracts, and contaminants for the purpose of source determination, characterization of depositional environments, maturity, biodegradation, and for correlation purposes. He has published over 400 articles with over 14,000 citations and has presented papers at numerous meetings and conferences. In addition, he has taught several petroleum and environmental geochemistry courses to many companies in Asia, Africa, Australia, and South America. Today, Dr. Phil will discuss environmental forensics, past, present, and future. There will be a Q&A session after the lecture, and you can submit your questions via the chat box, or you can just unmute yourself and speak. So with that, please join me and welcome Dr. Paul Philp. Thank you everyone for joining us today, and I hope you enjoyed the lecture. Dr. Philp, now you can take over and share your screen.
Okay. You don't see it yet, right?
Not yet.
I'm working on it. Why did this happen? Still not seeing it? Oh, okay. You see it now?
Yeah, good.
You got it?
Yeah.
Cool. Okay. And you can hear it as well, right?
Yes.
Okay. All right. Well, I was going to say good morning. I'm in Colorado, so that's why I'm saying good morning. I guess it's lunchtime where you guys are. But thank you for asking me to make this presentation. I won't spend much time going into my background, but as he pointed out, I'm an emeritus professor at the University of Oklahoma. I was on the faculty for almost 40 years, retired a few years ago, but still fairly active in the environmental field and teach a lot of courses on environmental forensics. And so in the relatively short period of time that I've got available here today, I want to sort of give you an overview of what we're doing or what is being done in environmental forensics. I find it very interesting because it's almost like sort of crime scene investigations in the environmental field. And basically what we're doing here is looking at the origin and fate of organic contaminants in the environment, be it in the air, soil, or water, and trying to find out who's responsible for these contaminants. This is different from what the EPA does. There's a big environmental group in the EPA, or that's what they're basically focused on. But their work is mainly a monitoring approach. I mean, obviously they want to catch who's responsible for these contaminants, but at the same time, the techniques they use, the methods, are fairly rigid. Whereas in environmental forensics, we are far more flexible. We don't have well-defined methods. We have very good methods, but there's a lot of flexibility involved in the approach. And of course, there are a lot of contaminants out there in the environments. A lot of them, they may not be introduced intentionally. Compounds like the PFAS, NTBE, PBDEs, and things like that, not always being introduced intentionally, but they're on a lot of products that we use, household products. They get disposed in landfills and things like that. And so they ultimately end up in the environment. I think if we, little bit of history here, interest in environmental forensics probably can be traced back, at least in the contemporary era, to the publication of the book Silent Spring by Rachel Carlson. She pointed out about the increase in pesticides in the environment, leading to thinning of birds' eggs, particularly the eagles in California, coming directly from the presence of the increase in concentrations of those pesticides. And once that was published, people started becoming more environmentally aware of the contamination in the environment. And at the same time, the late 1960s, early 1970s was a time when there were a lot of advances being made in environmental or in analytical techniques, particularly GC, GCMS, and later we started to see the use of stable isotopes. And also at the same time, within the petroleum industry, there was the development of this field referred to as petroleum geochemistry. And again, this advanced because of the emergence of these different analytical techniques, but it led to the discovery and introduction of the use of compounds referred to as biomarkers, and I'll explain what they are in just a moment, but they became very widely used for exploration purposes. But at the same time, it was recognized that these compounds, these same biomarkers, not only could be used for correlating oils with their potential source rocks, but they could also be used for correlating oils that have been released from a tanker or a pipeline with the contaminant in the environment. And I think from that point on, there was a significant increase in the number of environmental contaminants that were being discovered and being routinely analyzed by these analytical techniques that were being developed. The major questions that we ask in any forensic investigation are the number of questions are fairly small. You want to know what the compounds are or what the compound is. That's the first thing that you're always looking for, which sometimes is very straightforward. Sometimes it's a little more difficult. Then where did they come from? Did they come from a single source or multiple sources? And what you have to remember here is that in many cases, these contaminants that are being released into the environment are also mixing with the background signature that's there. And we're talking only about organic compounds here. And just think of all the naturally occurring organic compounds that are in the environment that can commingle with these contaminants that are being released. And one thing that we have to do is separate out the fingerprints from the natural sources versus these, if you like, anthropogenic sources. Then we also need to know if these compounds are degrading, because if they're degrading, then the fingerprints or the signatures from the contaminants that you see in the environment can look quite different from what they did when they were originally being released. And that's another question or problem that you have to take into consideration. And then the bottom line of all of this is to take this information and trying to figure out who is responsible for this release of the contaminant into the environment. Some cases that's easy, some cases it's more difficult. And also, just following on from that last comment, some of these contaminants or releases are very easy to see, like an oil spill. It's very clearly visible in the environment. Other cases, you don't see the contaminant. The compounds may be in the vapor phase, in the air, or in indoor air. That's another place where you get contamination. They may be water soluble, so you don't see them in the groundwater or in the subsurface. And some of these compounds may be coming from a single point source. Others may be coming from very diffuse sources, like the PFAS compounds are a big source of interest at the present time. But these compounds are everywhere. But one of the reasons for that is because they're very widely used in a lot of home products, personal products, fast food restaurants, and things like that. So these compounds are ending up everywhere. But every case is basically different and requires a slightly different approach. You're going to use the same techniques, but the way in which you use those techniques may be somewhat different. And you also have to remember that some of these contaminants may have been in the ground for 70, 80, or maybe 100 years and have undergone quite extensive degradation. Think about the coal tar deposits in industrial areas, probably like Pittsburgh or on the East Coast. A lot of coal tar deposits there from almost 100 years ago, or maybe more. DDT off the coast of California was dumped there back in the 1950s and was recently rediscovered, causing environmental problems there, still causing environmental problems. So in addition to all the analytical work that we do, you also have to do a lot of background or gather a lot of background and historical information, utilize aerial photos, satellite photos, and things like that. So it becomes a very comprehensive study, any of these cases that we're talking about. And obviously we can't cover all of that today. I can just sort of highlight some of these things. But typically any of these investigations will start off with some form of fingerprinting, typically gas chromatography. That's the basic doorway into these investigations. And this may give, hopefully will give some indication of what the nature of the contaminant is and whether or not it's been weathered. And by weathering here, we mean biodegradation, water washing, or evaporation. Those are the three major sources of weathering of these organic contaminants. And as I mentioned earlier, all of these processes can change the characteristics of the contaminant, resulting in a totally different fingerprint from the original product that was released. And if you have, with the groundwater contamination cases, if you have a single component, say MTBE or PCE, something like that, there are a whole lot of additional problems that you have to take into consideration, which I'll touch on a little bit just later on. The techniques, as I said, they're pretty standard these days. They're summarized here, quite a wide variety, but basically you've got some form of chromatography and some form of mass spectrometry. And in addition, now the stable isotopes are playing a much greater role in environmental forensics. Not going to go into all of these techniques, but that'll just give you an idea of the sort of things that we might be doing in any of these cases. And these are the same techniques that the EPA use, but we just tend to use them in a different sort of way. And we don't have to, or we don't adhere to these rigid analytical conditions that the EPA methods do. We don't have fixed detection limits. We don't have any list of target compounds. We may find that a compound that's just there in trace amounts becomes very important in being able to distinguish between two sources of contaminants. And, pictures like this, you've seen a lot of these in the past. Obviously, this is an environmental disaster here. And so another thing that's done is just not necessarily in a case like this, you're not necessarily going to pin down all the sources, but you're going to look at the impact of these compounds on the environment. Oil spills, of course, major environmental disasters. This was the first major tanker that ran aground back in 1967. This was off the coast of Cornwall, actually, where I was growing up at the time. This spill, if you quickly read what's at the bottom there, this spill was about 3 times the size of the Exxon Valdez spill. And very little information was available at the time about how to clean up a spill of this nature, of this size. And in the early days of this spill, as you see at the bottom there, they were trying to remove the oil by burning it. They were bombing the tanker. They were using napalm, gasoline to pour on the oil to get it to burn. Wasn't very successful. This is off the south coast of Cornwall. And a lot of this oil drifted across the coast or across the channel to the coast of France, and they got rather excited about it, and a lot of political problems as well as the natural problems. The Exxon Valdez, here's another big spill that I'm sure you're familiar with. This was back in 1989. Tremendous amount of forensic work done on this spill. A lot of good analytical chemistry came out of it. Techniques were developed, methods developed, which are still being used today. But what's of interest and worth mentioning here is that you see on the right-hand side that they were using steam cleaning or high-pressure steam to remove the oil from some of the beaches. The beaches in Alaska are pretty rocky, and they got the beaches clean, as you see on this slide up the top left. This is a beach that appeared to be cleaned up in 1989. The release was in 1989 as well. Looks very clean, but look in the bottom right. That's a beach up in Alaska where a lot of oil was accumulated. This is what it looked like in 2018. This is like 20 years later, and there's still oil that's there, and it's still there today and will be there for quite a long time, degrading naturally. And it seems that almost every decade, there's some environmental hot button that comes along. We had our DDT and pesticides in the 60s, oil spills, and a lot of these are still going on today, but this is when they sort of seem to peak in the literature. MTBE in the 80s, chlorinated solvents in the 90s, polybrominated compounds in the 2000s, and now we've got the PFASs, 1,4-dioxane, EDB, and things like that. I mean, these are just a few compounds. There are a lot more than this. And the PFASs, as I mentioned, are the big problem these days that you read about in the literature. And these are perfluorinated compounds. They're used in a wide variety of places: non-stick pans, stain-resistant furniture, waterproofing shoes, fire retardants, firefighting foams, and so on, and also widely used in food packing. And they're very resistant to biodegradation. This is just a slide that I found in the literature I thought was quite enlightening. This is where a study was done looking at different fast food containers. And you see there that in like those numbers, the percent numbers, what that's saying is that in the first one on the left-hand side, that in 56% of, I guess those are taco containers or wrappers, 56% of those contain these PFAS compounds. The paper cups they looked at, they didn't have any, but McDonald's French fry holders, 20%, and so on. So a significant amount of food containers contain these compounds, and they're in a wide variety of other sources as well. Okay, so that's enough of the little background and history, but let's get down to reality and see some of the problems that we encounter. As I mentioned before, once a release occurs and gets into the environment, then it could very easily mix in with either many other chemical products or the actual background in the environment. Can you see my cursor? Yeah, okay. So this is supposed, I mean, this is obviously a cartoon. This is supposed to be a river. What it's pointing out is if there's a release from some of these storage tanks over here into this, this river, then you've got the background from the plant material. You may have a jetty here that's got pilings with creosote on it. All of these things could end up being mixed together. And from a forensic point of view, we have to basically factor out these other contributions from the major contaminant source that was going into this river. And there are various things that we can do, various compounds that we can use, because there are certain compounds that we can associate with different background materials. For example, I won't go through all of this, but if you've got plant material, plant debris, then you have waxes on that debris, naturally occurring waxes. Those waxes contain normal alkanes that have a very pronounced odd over even distribution of normal alkanes. They contain stearines as well. Now, crude oils, for example, do not contain stearines, unsaturated stearanes. Crude oils only contain stearanes. So if you start finding stearines in your soil extracts, then you know you're looking at the plant debris. If you find stearanes, you're looking at crude oil material. And so we can eliminate different sources like that. Now, we're going to see several chromatograms in the next few minutes. Here's a chromatogram of a soil extract, and this is the C15 normal alkane, C16, C17, C18, and so on. Pristane and phytane isoprenoids are right here. But you see this in the early part of the chromatogram, you see this very smooth distribution of the normal alkanes. That's definitely coming from a crude oil source. But you look out here, you see we've got C27, C29, C31, a very pronounced odd over even distribution of normal alkanes. This is coming from a plant contribution. So this is an example of where we've had crude oil released onto a soil and mixing in with the hydrocarbons coming from the plant material. So you can separate those two out simply on the basis of the normal alkanes. Fingerprints of other commonly occurring contaminants are shown here. This is obviously not the complete set, but just a few of them. But you see, with some experience and knowledge of what to expect, it's pretty easy to distinguish gasoline, say, from condensate, condensate from jet fuel and diesel and so on. They have different distribution of hydrocarbons. These are all hydrocarbons. These are just the gas chromatograms. And this is fine as a starting point. The problem, of course, occurs when you may have multiple potential sources, say, of a gasoline release. You know, a lot of cities, you have crossroads, intersections where you've got gas stations on each corner. And there could be a gasoline release there. well, which one was responsible? You look at the chromatograms, they may all look identical, because it could turn out that all of the gasoline for those 4 gas stations has come from the same distribution point. And even if they come from different sources, they may have very subtle differences. So you have to look, well, before I go on to that part, here are a couple of gasoline chromatograms. And peak 24, for example, is toluene. These are xylenes. These are trimethylbenzenes. These are naphthalenes right at the end here. And now these two are not identical, but they're similar in terms of the distribution of compounds there. A big difference is that this gasoline, the one at the top, peak number 10 is MTBE. These are obviously older gasolines, but this gasoline has MTBE. Peak 10 is missing from this one. No MTBE in this gasoline. Very easy to distinguish those two. But in other cases, you may be looking for more subtle differences. All gasolines are going to contain alkylates, C8 compounds, which are branch compounds. The most common alkylate in gasoline is this 2,2,4-trimethylpentane. That's the compound that's got the ring around it over here. And these compounds, of course, are added to gasoline as octane boosters. And the distribution of alkylates that you see in different gasolines will depend upon where the gasoline is being produced, whether or not, or what type of alkylate production facility that particular refinery has, and how they've decided to blend the alkylates into the gasoline to adjust the octane rating to get it up to 85 or 96, whatever it is they're trying to do. Because you can adjust the alkylate in many different ways. You can have 2 gasolines which have exactly the same alkylate rating, but have a totally different distribution of alkylates and other compounds that are being added to adjust the alkylate rating. So again, a background knowledge of how these gasolines get their alkylate number can be very useful in distinguishing samples. Crude oils are a little more interesting in some ways, because crude oils, you can think of these as having their own sort of DNA fingerprint. Maybe not quite as specific as human DNA, but it's based on the concept of biomarkers. And these are compounds that are present in gasolines that are derived from functionalized compounds that were present in the original source material deposited in the environment hundreds of millions of years ago. And the biomarkers that we look at are hydrocarbons that are derived from these functionalized compounds. Let me go to this one first. So this is what we mean by a biomarker. We get sterols in the source materials for the crude oils. When these sterols are deposited in the environment, then they become a stearine. We know the type of source material a certain sterol is associated with, and so then we can say where this stearine came from, what type of source material the stearine came from. And we have a whole lot of families of biomarkers that have been developed by the petroleum chemists, and now we can use in the environmental areas. And the fingerprints from these biomarkers become very powerful tools for correlating, well, in the petroleum industry, oils with source rocks, in the environmental industry, correlating spilled oils with potential sources. Let me just go back to that one that I skipped over. Crude oils, very complex mixtures, of course. This is a crude oil. These are got the homologous series of normal alkanes. There are a lot of compounds in this early region of the chromatogram, which are compounds ranging from, well, basically from ethane out to about the C10 region. But we know what all of these compounds are. Of course, they're relatively volatile. So once an oil has been sitting around for any length of time, a lot of these compounds, the early eluting compounds, will disappear. But the semi-volatile compounds above C10 will remain. And these, on this slide, these are other families of biomarkers that we can use for correlating crude oils with spilt crude oils. I'm just, again, we don't have time to go into this in detail. This is just one example. These are all terpenes. These are tricyclic and pentacyclic terpenes, which elute in this region of the chromatogram. This is around C17 again. This is extending out to about C40. But we know what all of these compounds are, but in an environmental context, we just use this as a fingerprint. And if two oils have a similar terpene fingerprint, we're going to say, in all probability, they're related to each other, derived from the same source. These are stearanes. Again, very complex mixture of stearanes in crude oils, but very specific because these are coming from certain types of source materials. But it's another fingerprint that we can use. And on this slide, what I'm doing are showing the terpenes and the stearanes from 3 different crude oils. This is one crude, second crude, third crude. And I mean, obviously I picked the samples so they look that are very different so that you can see the extended differences that we might see between different crude oils. And none of these 3 would be related to each other. And again, we know from a petroleum perspective what all of these compounds are, and they provide a lot of information on the history of the oil. But from an environmental context, all we need to do is use these fingerprints for correlation purposes. And then here, when oils biodegrade, they lose the normal alkanes. And so here's a non-degraded oil, here's a heavily degraded oil, just the gas chromatograms. You look at those two, they look totally different. And there's no way, based on those gas chromatograms, that you can say those 2 samples are related to each other. But if we look at the terpenes and the stearanes for those 2 samples, here's the non-degraded oil, here's the degraded oil. You compare those fingerprints, and even if you look at them quickly, you can see that those fingerprints for the non-degraded oil and the degraded oil are basically identical. So these compounds do not degrade very quickly. They've been in those oils for 100 million, 200 million years. They've been sitting there happily in the reservoir. They're not degrading, and they're not going to degrade in a week, a month, sitting on the beach or wherever the oil is being released. Now, here's another example. There's not much information on that slide, but this is an industrial site in California. There was a big plume of diesel underneath this site. One company owned site A, another company owned site B. The 2 sites were divided by a railroad track, but they were arguing about who was responsible for this diesel plume underneath the site. And first of all, they were arguing whether or not it was a homogeneous plume or 2 separate plumes. Well, I'm just going to show you a couple of examples from the monitoring well here, the monitoring well there, and just give you a brief overview of how the story developed. These are the extracts from those 2 monitoring wells. And if you look at these 2 chromatograms, even if you don't know anything about diesel hydrocarbon distributions, you can see they look different. Well, there's a very good reason why they look different, and that's because this plume or this sample is partially degraded. If we look over here, this is C18, and that's phytane, which is the isoprenoid. You see how greatly reduced the normal C18 is, which is typical when something is being biodegraded. And down here, there's the C18, there's the phytane, a very much greater abundance of the C18. So the reason they look different is not because the plume is heterogeneous, it's because this part of the plume was partially degraded and this part was not degraded. So that's the first point to make here. The second point is that I mentioned early on how stable isotopes have become very important. Now, I know some people are more familiar with stable isotopes than others, and we don't have time to sort of go into detailed explanation here, but basically, stable isotopes represent the relative proportions, in this case, of carbon-12 and carbon-13. And they're expressed by a number. This is minus 21 per mil. That's the delta delta C13 number for this C14 isoprenoid, C15 isoprenoid, and so on. And if two samples are related, they will have similar isotopic values. And you see here, these are the two samples, the isoprenoids in those two samples again, and they're very similar, suggesting that in all probability, as they say in the courts, these two samples are related. But you don't leave it there, you take it another step. And these compounds, these are called adamantanes, very stable structures, cage structures, very difficult to biodegrade, so very resistant and persistent in the environment. And there we were using GCMS single-line monitoring to get the fingerprint for these adamantanes in these two samples. They occur as a number of different isomers, but you can see here that those two fingerprints basically identical. So what that's telling you is that in this case, that was one big homogeneous plume of diesel under that site. What can we do with gasolines? No biomarkers in gasolines. So one thing that we can do is to use the stable isotope values for all of these individual compounds to see if we can either correlate or distinguish between two samples of gasolines or three samples of gasolines. And let me skip over that one. And here's an example showing the result from obtaining the stable carbon isotope values. Back on that slide, I had the peaks labeled from 1 to 46. And then on this spreadsheet here, we're looking at the carbon isotope values for those 46 different compounds. And so we've got 4 gasolines here from 4 different locations. And you can see that in this particular case, we've been able to split those 4 gasoline samples into 2 groups of 2. Now this works so long as the gasolines are coming from different crude oil sources, where the crude oil sources themselves have different isotopic compositions. Doesn't work every time, but it's something that you can use. And with these forensic investigations, you're going to try and use as many different techniques as you can to get as much information as you can about these different compounds or different products to try and distinguish them. Another important thing is age dating. How long has the product been in the environment? There are many reasons for wanting to do this, and it's particularly important with gasoline. This is just one example of how this can be done, because over time, the composition of gasoline has changed due to changes from a regulation point of view, differences in engine design and things like that. And so here we see this ratio of toluene to 2,2,4-trimethylpentane to C7 plus C8. And you see over this period from 1970 to 2006, how around 1994, how the composition of gasoline changed quite significantly. And the alkane content in the gasolines was reduced. The aromatic content was actually increased and the alkane content decreased. But you see, if you find that you've got a gasoline where you've got a high value for this ratio, then that gasoline is probably younger than 1994. There are other parameters that you can use. There's a whole lot of information on here that we won't go into. We just don't have time. But additives, again, like MTBE, the alkyl leads, of course, were being used at different times. So we use a combination of these things to try and get a fairly accurate value for the age of the gasoline. The groundwater problems, this is huge. I mean, just about groundwater, wherever you are, is going to be contaminated with at least one, maybe more contaminants. And of course, a lot of times you don't see them. They contaminate the drinking water supplies and so on. A lot of them are very recalcitrant. They don't degrade very easily. But once you're doing one of these studies, you have to try and figure out where these compounds are coming from. And a lot of them, of course, are water soluble. So that also means that these compounds are relatively volatile. And so one way that's being used quite extensively now is, again, based on stable isotopes. And the thing is that since the 1980s, excuse me, since the 1980s, we've been able to measure the individual or the isotopic composition of individual compounds in groundwater samples. And now we have the capability to measure carbon, hydrogen, nitrogen, sulfur, chlorine, and bromine. Obviously, not every compound has all those different isotopes, but like many chlorinated compounds will have carbon and hydrogen and chlorine. So if you can measure 3 isotopes down to 1 ppb concentrations, then you've got a good set of fingerprints that you may be able to use for correlation purposes. Just a couple of quick examples here related to MTBE. Here's MTBE. Even though this was phased out many years ago, it's still present in the groundwater in a lot of different areas, still being produced in this country, being exported to South America. But this is it, methyl tertiary butyl ether. And so this can degrade either by removal of this methoxy group that you see here, or by removal of one of these hydrogens on the methyl group. And it depends the conditions of the environment where the MTBE is present. But this is an old map or old diagram, which is, I think, pretty interesting. This is a site at Lake Tahoe. Back in the late 1990s, there were big MTBE releases there. And this is one gas station. There's a drinking water well right here. This well was pumping drinking water, and the MTBE that was released from that gas station was basically being sucked over to this well and then contaminating the groundwater. They realized the groundwater was contaminated, the drinking water was contaminated. They stopped pumping, and then that MTBE started moving in the direction of the groundwater, the direction of the groundwater, where it encountered MTBE from another well, another gas station that had a release. So these two plumes commingled with each other, and then the forensic people working on this case had to basically figure out how much MTBE was coming from this site, how much MTBE was coming from that site using stable isotopes. Here's another quick example. This is a gas station in California, believe it or not, but these numbers are stable isotope numbers for the MTBE in these monitoring wells. And typically, MTBE will have an isotope value of around -28. Here you see a value of +38. Now, when compounds degrade, they become isotopically heavier, the numbers become more positive. So what that's telling you is that the MTBE in this region of the site is very, very heavily biodegraded. It's not degraded here at the leading edge of the plume. It's degraded in the middle of the plume. And with the MTBE studies, a lot of emphasis these days is placed on the extent of biodegradation, which you can follow through these stable isotope values. This is the same site, but basically, if you look at the color coding over here, you can see that it's estimated that in this area, over 95% of the MTBE has degraded, whereas at the leading edge of the plume, very little of the MTBE has degraded. I'm checking the time here. I just want to keep going, try to get through most of this. Now, chlorinated compounds in the groundwater, another big issue, far more complex than MTBE. But these chlorinated solvents like PCE, TCE, and so on, not only do they occur as the original compounds, but they undergo degradation. And when they degrade, they will end up forming, well, if we start off with PCE, that'll end up degrading to TCE, to the dichloroethanes, vinyl chloride, ethylene, and so on. You can end up with a very, very complex mixture of these compounds. And stable isotopes is the main tool that's used to unravel the source, to try and unravel the source of these compounds. And a couple of examples here. This is a site where there was a plume of PCE moving down towards this river. And there was a dry cleaner here that was thought to be responsible for that. And they put in a series of wells, monitoring wells right across here, and another series of wells down by the river. And used the stable isotopes to monitor the PCE. This is the results from the series of wells close to the dry cleaner facility. And that was very interesting because initially the dry cleaner was being blamed for everything. But once they got these isotope values for the PCE close to the dry cleaner, what was found was that there were, if you like, 3 distinct zones. One zone where the PCE had an isotope value of around -33, one area -27, one area -25. None of that is due to any degradation. So you can see that now the lawyers for the dry cleaner suddenly have a great opportunity here, because now it appears that maybe not all of that PCE was coming from the dry cleaner. Maybe there's another source there, or maybe 2 or 3 sources. Maybe somebody's dumped some PCE there. You know, all sorts of explanations that could lessen the amount of blame that's being placed on the dry cleaner. So you don't always get a perfect answer, but at least you can get some additional information. And then I think I've got a few more minutes left here. I've almost finished. But chloroform, this was a really interesting example. You know, chloroform, this was a study in Europe, a case in Europe. They found chloroform in some drinking water. And of course, the first solution is to blame some industrial source. But here what happened, and I'll just have to make this short, but basically they discovered the chloroform wasn't coming from, that was in this particular drinking water source, wasn't coming from an industrial source. It was coming from a natural source under a pine forest. And it turns out that a lot of chloroform is formed naturally underneath pine forests. And its isotopically is very different. The chloroform that you find under pine forests has a value between -13 to -27. Naturally occurring, or not naturally, industrial produced chloroform has an isotope value of -43 to -64. So that was a very interesting example and made people aware of another source of chloroform. And also, as an aside, termites produce tremendous amounts of chloroform. 15% of total global emissions of chloroform actually come from termites. What's that? Oh, and then there are 2 more slides here, I think. But just to give you an idea of where some of these things are going in environmental forensics, there's a lot of interest now in using ICP, inductively coupled plasma mass spectrometry, to determine isotopic compositions, not only of metals, but of isotopes like chlorine and bromine, oxygen, sulfur, and so on, because all of those isotopes show up in a lot of environmental contaminants. And you can get very good isotopic data using, this is just a little cartoon where we've got a GC coupled onto a ICP instrument. And there you can determine very accurately any of those isotopes. This is just one example showing a chromatogram of sulfur-32 isotopes. This is actually, I think, from a rock extract, but benzothiophene. You see, all of these compounds are different individual benzothiophene. And if you look at these numbers here, these are the sulfur isotope values. They're different for these compounds. And so now we have a sulfur isotope fingerprint that we could use for correlation possibilities. Excuse me, do the same thing with bromine and so on. So I think we're going to see a lot more of ICP-MS isotope data in the literature in the forthcoming years. So that's a little summary of what some of the things that we do in environmental forensics. Obviously, a wide variety of scientific disciplines there. If you want to get into this, a good knowledge of analytical chemistry is a good thing to have. Good understanding of how these compounds are being produced is also pretty useful, so that you can find little pieces of information that may enable you to distinguish a compound or a product coming from one source versus another. And of course, bear in mind that a lot of this ends up going to litigation. So the chances are you end up giving evidence in court, and you have to try and explain some of this scientific data to a jury. And remember, a lot of juries are comprised of people that just didn't have a good excuse to get out of jury duty, and probably have not got much more than a high school education and wouldn't have a clue what a stable isotope is. So try imagining explaining that to them in a few words. So I'll leave you with that. Hopefully it's given you some information and ideas of what we do, and found it of some interest. Should I leave my screen? I guess I should.
Thank you, Dr. Phil. We have time for a few questions. Okay.
Do you hear me?
Yes.
Okay.
Yes.
I pressed the wrong button.
Yeah, so you touch base on the biomarkers, okay? So how were the biomarkers selected? Like if we don't know the compounds, there's unknown compounds in the mixture, and how do we select the biomarkers? And what are some of the criteria for this selection?
Well, it's sort of very interesting the way that this all evolved, because again, this started back in the 1970s, and a lot of the early work was done mainly by oil companies, primarily by Chevron in the US and BP in England, and a small number of academic groups. And basically, it's also tied in with studies that were done on, and this may sound weird, but studies that were done on lunar samples and Precambrian samples, where people were looking for compounds that were thought to be markers for indicators of life. And they started finding stearanes, for example, and terpenes in these very old rocks. And they were identifying what these compounds were, but then some of these compounds were being found in crude oils. And so once we got an idea of what some of these compounds might be in the oils, then those sort of families of compounds expanded into other families of compounds. And over time, we sort of built up large groups of compounds that we could actually associate with different living sources of organic matter, and then figure out what the precursors of those compounds were. I mean, obviously in the case of stearanes, it was fairly easy. You know, you see a stearane structure, and then you know that that structure is coming from a sterol. And then there's a lot of natural product literature that tells you what different sterols, what sources of organic matter different sterols are associated with. And so you start putting this together and building up this, if you like, family of biomarkers, build up these families of biomarkers that you can use. And, you know, that started, as I said, well, it actually started, I've left out an important step here, because a very famous German chemist, a guy called Alfred Tribes, back in the 1930s, discovered the structure. He discovered the structure of chlorophyll, and he then figured out that in crude oils there were compounds called porphyrins, and he figured out that porphyrins have very similar structures to chlorophyll, and he made that relationship between those structures, And that was really the very first biomarker that was ever discovered. And then he did that using very classical chemical techniques. And then once the GCMS came along, it was much easier to detect and identify these other compounds.
All right. You show one example that in California, there's two sites, A and B, and it turned out one side there's a biodegradation, right? And the other side is not. So with this kind of information, is it possible to better understand like the biodegradation mechanism, why that happened in one side but not in the other side?
Yeah, in a lot of cases it is, because I mean, none of these plumes are necessarily homogeneous, and there's variation in the subsurface, primarily in terms of the oxygen content, the redox potential, and some parts of the plume may be very highly reducing, other parts very highly oxidizing, and that's going to be one of the prime factors that will determine the rate of degradation. And it's all, when you've got a hydrocarbon product like diesel or crude oil, the compounds that go first are always the normal alkanes, and they degrade most rapidly under oxic conditions. So that part of the plume was highly oxic. The other part was more anoxic, making it slowing down the rate of degradation.
So from your presentation, it seems like the analytical chemistry, especially the instrumentation, will continue to play a critical role in this field, like ICP mass spec, GC mass spec, and others. But recently, like we know, the AI and machine learning is a very hot topic. Like everyone is now talking about this. So what's your perspective on the role of AI and machine learning which will play in this field?
Well, yeah, it's sort of interesting to see if it's going. I mean, I'm sure it'll play a role somehow or other. But I mean, I think even if it does play a role, we're still going to have to check the data, because, you know, the data, as I said at the end there, a lot of these cases go to litigation. I mean, a lot of them are settled out of court as well. But I think, you know, if you just depend upon the sort of AI view of reaching a conclusion, you could be wrong. I mean, you may be right as well, but I think you're going to still need that human impact to decide, or input to determine whether or not you really have reached the right conclusion. I mean, maybe it'll reach the point where people become so confident in it that they don't bother to do that anymore. I don't think that'll be in my lifetime. But again, I guess you're also going to run into the situation where people are going to use AI and just make a totally incorrect interpretation and say that it's correct. I think there's a lot of room for fraud or misinterpretation there. So I don't know. I think you just have to be cautious.
Right. Yeah. All right. It's 1 p.m. now. I think it's time to close this lecture, and let's thank our speaker again, Dr. Philip. Thank you very much. Very nice talk and very nice discussion. Thank you.
All right. Thanks very much.
All right. Thank you, everyone, for joining today. Thank you. Bye-bye. Have a good day.
Bye.
Thanks. Bye-bye.
Bye.